Welcome back. We've talked about how we can take organisms and put them into groups based on their evolutionary relationships. And for most of the remainder of the course, what we're going to be doing is looking at those groups, looking at groups of animals and plants and fungi and bacteria and so on, and figuring out what special characteristics define those groups and what makes each of those groups pretty amazing. So we'll look at the diversity of life on Earth. But before we do that, we're going to look at a group of things that are not generally considered to be alive. Most biologists would not consider viruses and prions, another thing we'll talk about that maybe you haven't heard of, to be living things, even though they reproduce. And in the case of viruses, they have a genetic code, but they can't reproduce on their own. They don't have a metabolism. They can't do most of the things we associate with life by themselves. Instead, they have to hijack cells and take over the machinery of those cells. So a virus, a biological virus, is a lot like a computer virus. Think about a computer virus. It really can't do anything if it's just sitting on a flash drive or if it's on some sort of storage device and that device is turned off. It can't do anything. It needs an operating system. That operating system will be tricked into making copies of the code and perhaps spreading that virus to friends and family on your contact list. Biological viruses are much the same. They contain code. There's a packet made of protein, and inside that we have a code of nucleic acids. That would be either RNA or DNA. And what happens is the virus injects its code into a cell, and it uses the same code, so that cell will be tricked into copying that code and also using that code, decoding it to make the protein coat that protects the genetic information and helps the virus invade a new host cell. Before we get to this, though, we really do need to spend a little bit of time looking at how DNA and RNA, genetic material, is used within cells. So we'll start off by looking at, I suppose, the operating system of life. Molecular biology, as the name suggests, is the study of molecules in living cells. But specifically, it tends to focus on heredity, genetics, how DNA serves as genetic material, serves as code or information, and how that DNA codes for proteins. Proteins do all the work in the cell. So in eukaryotic cells, like the cells that we're made out of, we have a nucleus, and the nucleus contains DNA. And a lot of people have probably suggested that the nucleus of a cell is like the brain of a cell. I don't really like that analogy. The nucleus is like a library. It contains information. So within the nucleus, we have bookshelves, and those bookshelves are like chromosomes. You have 23 chromosomes that you got from your father and 23 from your mother, and those bookshelves contain information. The individual books on the bookshelf, and each bookshelf will have on average about 1,000, those books are genes. A gene is a sequence of DNA, and that sequence is the instructions needed to make one specific protein. So again, I think of the nucleus as a library, bookshelves are chromosomes, books are genes, genes code for proteins. So the DNA is a fairly stable molecule. It's going to stay within the nucleus, and every time a cell divides, the DNA is going to be copied and passed down to the next generation of cells. But the DNA can also serve as a template to make something called messenger RNA. And that's exactly what it sounds like. It's a messenger. So this is a single strand of nucleotides, and we'll talk about that in a second, that carries the information from the DNA. The DNA is not moving, it's staying put. It carries the information, a copy of it from DNA, to the cytoplasm. And in the cytoplasm, we have ribosomes. And ribosomes bind to that messenger RNA, and they use it to take amino acids and put them in the proper sequence to make a specific protein. So DNA can replicate, it can make copies of itself, but it can also be transcribed into messenger RNA, and then the messenger RNA can be translated to make proteins. And if this isn't making sense just yet, don't worry, we're going to go over each of these steps. The large molecules in your cells are made out of much smaller building blocks. And you have 4 groups of large molecules. You have fats, also known as lipids. You have nucleic acids, which are the RNA and DNA molecules. You also have proteins, and you have carbohydrates. And each of them, like I said, is made out of small building blocks stuck together. Think of it like building something out of Lego. If you're taking amino acids and sticking them together like Lego blocks, you're building a protein, for instance. And if you take what are called nucleotides and you stick them together, then you build a nucleic acid. When these nucleotides are used to build a molecule, once they're in that molecule, we refer to them as nucleosides, and there's 3 parts. We have what's called a phosphate group, and we have a sugar, and that could be ribose in RNA or deoxyribose in DNA, but this phosphate and this sugar make up the backbone of the molecule. And then we have a nitrogenous base. So we have these 3 parts on each nucleoside, and these nucleosides are strung together in long chains. The nitrogenous base, and it's called that incidentally because it contains a lot of nitrogen, but the nitrogenous base can be one of 5 different types. In DNA, we have adenine, thymine, guanine, and cytosine. And to keep things simple, we usually abbreviate that as A, T, G, and C. And then in RNA, we can have adenine, guanine, cytosine, or uracil. So notice that we don't have the thymine there, we have uracil instead. But just with these 5 different nucleotides, just with an alphabet of 4 letters, if we think of the genetic code as a language, with an alphabet of 4 letters, we can code for everything that your cells need to do and all the programming that brings your cells together to form you. Let's start by taking a look at these 3 representations of a DNA molecule. And we'll start with the one in the middle. Notice that we have 2 chains of nucleotides. This is a nucleotide. We have a phosphate, we have a sugar. In this case, it's going to be deoxyribose. We'll come back to that. And attached to that sugar, we have a base. And this particular base is adenine. Now the phosphate and also the sugar link together one after the other. So we have a phosphate, a sugar, over and over and over again. And that forms the backbone of the chain. In the first diagram, you can see those backbones, the phosphates and sugars, represented as a blue ribbon. And notice these 2 chains twist around each other. Let's have a look at that base though, because the base is the important part. That's the code. Looking at the sequence from the top on that strand, we have thymine, guanine, cytosine, and adenine. Notice that on the opposite strand, we have a complementary sequence. And what I mean by that is if there's a T, we have an A on the other side. If there's a G, a guanine, we have a C, a cytosine on the other side. We always have this pairing. So T always binds with A, and G always binds with C. Those little red dots between the two strands, those are hydrogen bonds. Now this is a rather weak bond. Each hydrogen bond is quite weak. It's just due to a slightly different charge on one side versus the other side. So one side is slightly negative, one side is slightly positive. Opposites attract. But each of those hydrogen bonds is weak and fairly easy to break. And this is important because this means the two strands can be unzipped. They can be pulled apart just a few nucleotides at a time, and it doesn't take a whole lot of effort from the cell. So again, the very first image there on the left is just showing the phosphates and sugar as a blue band, because that's just the backbone. It doesn't contain any actual information. And then it's showing the nucleotide sequences because that is important. On the right is what's called a space-filling molecule. This is showing the actual atoms as they might look, overlapping their shells, interconnected. So this is somewhat close to what a DNA molecule might actually look like if we could see it. Let's have a quick look at how RNA and DNA are different. Don't get too wound up in the structures here. The first difference is the name, of course, of the molecule, and that comes from the name of the sugar. So RNA is made with the sugar ribose. Notice that I've highlighted an oxygen and a hydrogen there. Now let's compare that to DNA. The sugar in DNA is deoxyribose. It's exactly the same as ribose, but there's an oxygen missing at that same spot. D means without, so without oxygen, ribose without an oxygen. If you want to be really specific, it's 2-deoxyribose because it's found on the second carbon. But we're not going to get into that level of detail here. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. There are two additional things that separate RNA from DNA. One we've already talked about. So in RNA, we have a base known as uracil, and in DNA, we have a very similar base known as thymine instead of uracil. In cells, RNA is single-stranded, whereas DNA is double-stranded. So RNA is just one strand of nucleotides, whereas DNA is two strands. Two strands of nucleotides held together weakly by hydrogen bonds. I just want to point out, though, this is the way it works in cells, but viruses can do things a bit differently. There are some viruses that have double-stranded RNA, and there are some viruses that have single-stranded DNA, as we'll see. Let's have a look at how these molecules are used in cells to store and transmit information. RNA is a rather fragile molecule. There's lots of enzymes wandering around in cells that will gobble it up if they get the chance. Really, you just have to look at an RNA molecule funny and it will break apart. DNA is more stable. Those two strands help protect it. It helps protect the sequence of information from being degraded and broken down. But DNA, as mentioned, can be unzipped. So the hydrogen bonds between each pair of nucleotides are fairly weak, and they can be unzipped a little bit at a time. And that gives the cell access to the information, which is the chain, the specific sequence of nucleotides within the DNA molecule. DNA can serve as a template to make copies of itself, to make copies of the information it contains. Whenever a cell is getting ready to divide, the DNA has to be duplicated first. So when a cell is finished dividing, both of the daughter cells will both have a set of information. The enzyme that's responsible for copying DNA is known as DNA polymerase. And what it does is it unzips the two strands of DNA a little bit at a time to get access to the genetic code, read it, and make a complementary strand. So as you can see here, we have TCAG going up in that region of replication. And DNA polymerase would grab onto the complementary nucleotides A, G, T, C and put them together. And we end up with two strands that are identical to the parent strand. DNA can serve as a template to make another copy of DNA. Of course, that has to happen so that cells can maintain information and pass it down from generation to generation. But we haven't talked about how that information is decoded and used to make proteins, because remember, that's the ultimate goal. There's two steps. The first step is known as transcription. The second step is known as translation. I like to think of nucleic acids as a language, and it's a language with only 4 letters. So DNA has 4 letters, 4 nucleotides, and RNA has 4 letters, 4 nucleotides. Proteins, different language, they have an alphabet of 20 letters. And those 20 letters are the 20 different amino acids that are found in your cells. The first step, transcription, is performed by an enzyme known as RNA polymerase. Incidentally, a polymerase is an enzyme that takes little pieces and puts them together to form a big molecule. So you have DNA polymerase that makes DNA, RNA polymerase that makes RNA. RNA polymerase will latch on to a piece of DNA. Specifically, it latches on to the beginning of a gene. And what it does is it opens up the DNA a little bit at a time. It travels along it. It reads one strand of DNA and uses that as a template to make messenger RNA, which will go on to the next step. So you can see here, RNA polymerase sees a T, it puts an A. It sees an A, it puts a U, and remember that's because RNA has U's instead of T's, etc. It reads along. When it gets to the end of the gene, it spits out this messenger RNA, and then that will go on to the ribosome and to the next step, which is translation. Ribosomes are the smallest organelles in the cell, and what they do is they grab on to messenger RNA that's left the nucleus, and they decode it. And they use the code to put amino acids into a chain in the correct order. The order of those amino acids is going to determine the structure of the protein, and that's going to determine what the protein does. So, on the messenger RNA, the code is read as triplets. And what I mean by that is that 3 nucleotides are read as a word, basically. And those 3 nucleotides are known as a codon. So we have these words made out of an alphabet of 4 letters, and the words are 3 letters long. They're known as codons. Each codon codes for a specific amino acid. There's 20 amino acids in the cell. So for instance, AUG, this codes for the amino acid known as methionine. It also tells the ribosome where to start. AUG is referred to as the start codon. CUA codes for leucine, GGC codes for glycine, etc. Humans cracked the genetic code back in the 1960s. And what's most fascinating about this code, I think, is that it's universal. What I mean by that is all living things use the same code. Giraffes, bacteria, oak trees, humans, doesn't matter. All living things and viruses use this same code. And that's because we can trace it back to the very first organisms, the first cells that appeared on the planet. So this is an ancestral trait. Now, we can decode any sequence of messenger RNA using this decoder ring that we see here. And it's a lot like a secret decoder ring that you might use to decode a secret message. Let's take a look at how it works. In any sequence, there's going to be one codon that the ribosome recognizes. So in a messenger RNA, there's a bit of junk at the beginning and a bit of junk at the end. But the ribosome looks for the codon AUG. And that's how it knows where to start. That's how it knows where the actual information begins. So on this decoder ring, we start in the middle. Our first nucleotide is A. Our second nucleotide is U. And our third one is G. And this codes for the amino acid methionine. Now it's also the start codon. Again, it's a really important codon. It's the one that the ribosome uses to line itself up. Let's take a look at another codon. Let's say we had the codon UCA. We'd start in the middle with our U, our uracil, and then our C, and then our A, and this would code for the amino acid serine. Now, one other thing I want to point out, we have 20 amino acids. We have one, the one for methionine, that's also a start codon, but we also have stop codons, and you can see those here. There's 3 ways to code for those: UGA, UAA, and UAG. And those stop codons don't code for an amino acid. Instead, they tell the ribosome where to stop. Basically, there's a protein that binds there and it causes the entire ribosome to fall apart and leave the messenger RNA. To summarize our quick little discussion of molecular biology, DNA serves as a template to make more DNA, and the process is accomplished by DNA polymerase. In eukaryotic cells, the DNA is going to be found in the nucleus, and it's going to stay in the nucleus. Then we have transcription. Transcription is where DNA is used as a template to make messenger RNA, and this is done by the enzyme RNA polymerase. That messenger RNA can then leave the nucleus. There's nuclear pores, these little holes in the nuclear envelope that it goes through, and it goes out to the cytoplasm and it finds a ribosome. Now finally, at the ribosome, we have our messenger RNA being decoded, and that code is used to make proteins. And again, proteins are just chains of amino acids, but the code that's found in DNA and RNA is ultimately used to string together amino acids in the correct sequence. Now we have enough information here that we can start to talk about what viruses are and how they work. Viruses take over this process, or they interrupt this process. Viruses can contain DNA that's used as genetic material or RNA that's used as genetic material. So quite different from what happens in cells. Cells always have both DNA and RNA. Viruses have one or the other. Let's have a look at the general structure of viruses. Let's take a quick look at the size of viruses. They are incredibly tiny. So tiny that it was quite a long time before we discovered them. We discovered cells much earlier. So cells can be seen with a light microscope. In this diagram here in the back, you've got a liver cell, which is a pretty typical human cell. And you can see at this scale, we're just seeing a tiny sliver of it. In front of that, we have E. coli, a pretty typical bacteria cell, but the viruses are much smaller. With a light microscope, you might see a large virus as just a dot. If you want to see any more detail, you need to have an electron microscope. So quickly going through some of these viruses, because they are ones that are rather important. Some of them you will have heard of before. We have a pox virus there. Of course, very important historically. Within that group of viruses, we have smallpox and cowpox and a few other things. Smallpox is the one that has been most important. It's a very, very deadly virus. Of course, it's the virus that wiped out a large percentage of the natives in North America, unfortunately, when the Europeans first arrived. We have the herpes viruses. This is a big group, causes things like genital herpes and cold sores, also responsible for chicken pox and Epstein-Barr and a few other things. We have T4. You might not have heard of that one. This is a bacteriophage. A bacteriophage is a virus that infects bacteria. And this is one of the best studied viruses because it's easy to study in the lab. We have the polio virus. Was a very deadly virus, still is around. It's found in small pockets of Africa and so on. It was very, very important throughout the world in the last century, in the 1950s, and still continues to be important. Like I mentioned, it's not completely being eradicated, although we do have good vaccines against it. Adenoviruses, this is one of the groups of viruses that causes the common cold. The common cold can be caused by a lot of different viruses, as we'll see. TMV, now you might not have heard of that. This is the tobacco mosaic virus. It infects tobacco plants and causes them to be discolored and unhealthy. And you might wonder, well, why is that important? This is actually the first virus that was discovered and studied in detail. We have the influenza virus, which of course causes the flu, which is one of the most common viral infections and does actually kill quite a few people. Here we have a generalized schematic for a generalized virus. In the middle of the virus, we're going to have genetic material. Again, this is the most important part of the virus. This is what's going to be injected or absorbed into the cell, and it's going to be replicated and decoded by the cell. This can be DNA or RNA, and as we'll see, it can be single-stranded DNA, double-stranded DNA, single-stranded RNA, or double-stranded RNA. But we're just going to have this one type of genetic material. One of the ways we can distinguish between the different families or categories of viruses is what type of genetic material they contain. Around that, we have a layer of protein, this protein coat or capsid that will contain and protect that genetic material. And for some viruses, that's the end of it. Other viruses will have an envelope around them. So enveloped viruses contain a membrane or envelope, which is a bit of plasma membrane that was stolen from the last host cell. And within that membrane, typically we're going to have viral glycoproteins. Remember, a glycoprotein is protein with sugar residues added to it. And typically these proteins are going to be used to attach to the plasma membrane of a host cell. So they're going to be used to enter a cell, and then as we'll see, they also play a role in exiting the cell. So a virus is a pathogenic entity that can infect a cell and hijack the genetic machinery of that cell. Now I'm going to throw a new term at you that you may not have heard of before. That's virion. Virion refers to the complete form of a virus, the form a virus takes when it's outside of a cell. So this is the form that can infect a new host cell. It's going to consist of genetic material, so DNA or RNA, surrounded by a protein coat or capsid, and depending on the virus that we're talking about, there may also be an envelope around that. Now that is the form that can attach to another host cell and get into that host cell. And when it does that, it's going to lose its envelope, and it's also going to lose the capsid. And then that naked genetic material is going to be replicated and decoded, so transcribed and translated to make proteins. You can use the term virus to refer to any part of the life cycle of that entity, but virion refers specifically to the complete form of the virus that's found outside of the cell. What you're seeing here are three different virions. First, on the left, we have a bacteriophage. This is a virion that can infect bacteria cells. And then we have two examples of enveloped viruses. So first of all, we have our capsid, our protein coat. And you can see in many cases it has kind of a geometric shape to it. Within that, we have our genetic material, our nucleic acid. That can be DNA or RNA, and as we'll see, it can be single-stranded or double-stranded in both cases. And then we may or may not have an envelope that surrounds that capsid. So we've talked about the structure of viruses. Now let's talk a bit about how they function. So viruses are essentially tricksters. They will trick a host cell into replicating their genetic material and also decoding it to make proteins. What's more is the cell will be tricked into taking those proteins and using them to package that genetic material. So the end result will be a mature virion that can then leave the cell and infect another cell. What you're seeing here is a fairly typical enveloped virus. This is the influenza virus. In the center, we have single-stranded RNA molecules, and each one is surrounded by a helical layer of proteins that make up the capsid. Around that, we have the envelope, and within the envelope, we have a number of important proteins that stick out on the surface. There's two of them that you've probably heard of before, or at least you've heard of the abbreviation. We have N proteins and we have H proteins. The N protein is used to attach to a host cell and trick the cell into taking in this virus particle. The H proteins are used to exit the cell. So you can imagine that these are pretty important proteins, and if they change in any way, that's going to affect what cells they're able to infect. Viruses tend to mutate fairly rapidly, so they can give rise to new strains on a fairly short timescale. And what typically happens is we have changes to the genetic code that will affect the structure of these N and H proteins and result in influenza strains that have unique characteristics. They might be able to infect cell types that they couldn't before, or they might be able to infect organisms that they couldn't before. You've probably heard of H1N1, which is a particularly virulent strain of influenza. And the H1 and N1 refer to the particular forms of H and N proteins that that particular strain possesses. Viruses are quite often very specific when it comes to what species they can infect and what tissue types or cell types they can infect. And that's because viruses will bind to surface proteins. And quite often, the proteins that they select, that they've evolved an affinity for, are specific to certain organisms or certain cell types. And you're seeing an example of that here. We have T lymphocytes, so part of your immune system, and we have HIV binding to proteins that are specific to those cells. Let's take a look at the life cycle of a virus. And we'll start with the bacteriophage because they are a little bit easier to understand. They're also very easy to study, so they're quite easy to raise in culture in a laboratory setting. So once again, we have our capsid that contains DNA, and then below that we have a sheath that connects the capsid to a base plate and tail fibers. The tail fibers are used to attach to proteins on the surface of a bacterium, and the base plate is used to basically inject DNA into the cell. On the right, you're seeing a scanning electron micrograph of a poor little E. coli that is covered with T4 bacteriophages. What you're seeing now is a bacteriophage, quite often just called a phage, that's going to infect an E. coli bacterium. It's landing on the cell. It looks like a little robot or a little spaceship. And it will identify proteins that are normally found on the cell and latch onto them. Now the next thing that happens is it inserts a drill-like structure and it punches a hole into the cell wall. The cell wall is made of something called peptidoglycan, and it inserts an enzyme that breaks down that peptidoglycan and punches a hole in the cell wall. Finally, it's going to insert this tube. It's hollow in the middle, and that will inject the genetic material, the DNA, into the cytosol, the liquid portion of the cell. Once a bacteriophage is inside a host cell, there's two different pathways or cycles that it can use to reproduce. The first cycle we're going to talk about is known as the lytic cycle. So again, the very first thing that happens is we have attachment of the phage to the host cell. So the tail fibers are going to stick to specific proteins on the outer membrane. Next, lysozyme is going to be injected into the periplasmic space. Lysozyme is an enzyme that breaks down peptidoglycan. We produce that same enzyme. We secrete it onto the surface of our skin, for instance, to limit the growth of bacteria. In this case, lysozyme is going to essentially make a hole in the peptidoglycan layer. That's going to allow a tube, kind of like a hypodermic needle, to punch through the wall and punch through the inner membrane and allow the phage to inject its DNA into the cytosol of the cell. Now the cell is going to start transcribing and then translating this viral DNA. And it's going to code for proteins that make up part of the capsid and also for some of the enzymes that the virus uses during this infection. And some of those enzymes are actually going to break down the chromosome of the cell. So now this cell is completely focused on reproducing, copying, and transcribing that viral DNA. Now, even though the DNA of that host cell has been broken down, everything else is still intact. So that cell still has active DNA polymerase, RNA polymerase, active ribosomes, etc. And this cell will begin to decode that viral information. And it will start to use it, of course, to make proteins. It will copy the DNA and package the DNA into capsids using the instructions that were provided by the viral genome. And these bits and pieces will be assembled into mature virions. Now another thing is happening. One of the genes that was transcribed and translated by the host cell was the gene for lysozyme. And that lysozyme is going to punch holes in the cell. And that will bring about the release of the active virion. So we can start off with just one virion injecting its genome into a cell, and we can have 200 or so virions leaving the cell and destroying and killing the cell in the process. As you might imagine, a viral infection like this can spread very rapidly from one cell to the other, and it can decimate a population of bacteria quite quickly. In fact, there's a number of researchers that are looking into manipulating these bacteriophages to make them more virulent in order to make them more problematic for bacteria that cause harm to us. The lysogenic cycle is even sneakier. So it starts out the same way. We have a phage that will attach itself to the outside of a bacterial cell and inject its DNA. But what happens with that DNA is quite different. The DNA is going to be integrated into the chromosome. And at this point, we refer to that piece of DNA, the entire genome of the virus, as a prophage. Now, the cell hasn't been harmed. It can repair that little hole that was punched in the cell wall and go about its merry way. And it will keep doing what bacteria do, which is divide. E. coli, for instance, divide every 20 minutes or so. But every time it divides, it's going to replicate that chromosome, including the prophage. So let's say we had one E. coli cell that was going to be infected with a virus, with a bacteriophage. If we were to take that infected cell and grow it up on a Petri plate, the next morning we would have billions of cells that were descended from that infected cell, and they would all have the prophage in their chromosome. Once again, though, no harm has been done to the bacteria yet. But there's a trigger of some sort that will occur eventually, and it might be a change in pH or some other kind of stressor that will cause that prophage to excise or cut itself out of the chromosome. And at that stage, we enter into a new lytic cycle. So now the chromosome is going to break down, and that DNA is going to be copied and transcribed and translated, and the cell is going to fill up with virions, and it's going to be lysed or ruptured. The two cycles are linked. However, only the lytic cycle causes damage to the cell. So if you have a virus within your cells that's in the lysogenic cycle, it's lying there dormant. We don't have any active virions that can cause damage. Instead, we just have information. That prophage will only give rise to the harmful form of the virus, the virion, if induction occurs. This sort of thing happens in humans as well. There's a number of viruses that will insert their genome into your chromosomes. For instance, there's a number of herpes viruses that do this. There's a herpes virus that causes chickenpox. If you get chickenpox as a child, damage is being done to your skin cells during the lytic cycle for that virus. But the virus will also incorporate its genome into nerve cells deep in the skin. Later in life, you can develop shingles, and shingles is the result of induction activating those prophages and causing them to give rise to new active virions. We've talked about how viruses can be categorized based on how they enter cells and their structure. But the most important criterion for organizing viruses into some system that makes sense is looking at the type of genetic material they have. So do they have DNA or do they have RNA? Is it single-stranded DNA or double-stranded DNA? Is it single-stranded RNA or double-stranded RNA? And if they are single-stranded molecules, are they the template strand or the non-template strand? Don't worry, this will all make sense soon enough. When it comes to the universal genetic code, viruses play by the rules. They have to, otherwise they wouldn't be able to do their tricks. They have to use the same code that the cells they infect use. However, when it comes to the central dogma of molecular biology, there are some viruses that bend the rules quite a lot. So remember that the central dogma states that DNA is a permanent storage of information. And DNA can be used as a template to make copies of itself. It can be used as a template to make messenger RNA. And messenger RNA is going to be used by a ribosome to make protein. And there are several viruses, DNA viruses, that follow these rules. But there's RNA viruses that have nothing to do with DNA. They don't use it at all. They use RNA to store their information, and they use RNA as a template to make more RNA. Even stranger, there's RNA viruses that use the RNA as a template to make DNA, and then they can insert that DNA into your DNA. That process of going from RNA to DNA is known as reverse transcription, because it's the reverse of the usual transcription, DNA to RNA. The viruses that do this are known as retroviruses, retro meaning backwards. Now also, remember in cells, DNA is always going to be double-stranded and RNA is going to be single-stranded. Well, there are some viruses that use single-stranded DNA as their genetic information. There are some viruses that use double-stranded RNA as their genetic information. So viruses are kind of all over the place. And now we'll have a look at the different techniques, the different genetic pathways that these different families, these different groups of viruses use to infect cells. What you're seeing in this figure is transcription. So we have a short piece of DNA that's being unzipped, and the top strand of the DNA is being used as a template to make an RNA transcript. RNA transcript, that's another name for messenger RNA in this case. Remember, it's RNA polymerase that's doing this. That top strand that serves as a template can be called the template strand. And we could call the bottom strand that's not being used the non-template strand. That makes sense, but that's not the terms they're using here. They're calling that upper strand, so the template strand, they're calling it the antisense strand. And that bottom strand that's not being read is being referred to as the sense strand. Now, why is that? Well, have a close look at the sense strand. Even though RNA polymerase is not paying any attention to it, the sequence in that sense strand is the same as the sequence that's being spat out in RNA by RNA polymerase. So down the bottom in the sense strand, we have T, A, C, T, G, C, C, etc. And in the messenger RNA, we have U, A, C, U, G, C, C, etc. The only difference between the sense strand of DNA and the messenger RNA when it comes to sequence is that, of course, in messenger RNA, we have U's instead of T's. But that messenger RNA can be referred to as sense RNA, and it has the same message as the sense strand of DNA. The RNA is referred to as sense RNA because the ribosome can make sense of it. It's the message that will be used to make protein. Out of the many varied groups of viruses, I have selected 3 that we'll talk about in a little bit more detail. And I've selected them based on what they use as their genetic material. The first group we're going to talk about are the double-stranded DNA viruses. Double-stranded DNA is what we use in our cells as our genetic material. A good example would be the herpes viruses. They will fuse with your cells and they get taken in, and their double-stranded DNA is dumped into the cell and it makes its way to the nucleus. Now, once it's in the nucleus, it has access to DNA polymerase. That's where it hangs out. And the DNA polymerase will make copies of that genetic material. It will copy the entire genome of the virus. The genome is the entire set of instructions needed to make the virus. In some cases, that DNA can become incorporated into your DNA. So as mentioned before, you can get chickenpox. The DNA is incorporated into DNA of your cells, and it can sit there for decades. And then many years later, it can pop out and give rise to active herpes virus again, this time causing shingles, which is more serious. Okay, so to try and wrap our heads around this, I've come up with this hypothetical sequence here, and I'm going to use this for each of the groups of viruses that we talk about. So let's say this represents a gene from the virus. Of course, a gene would be much longer than this. We have double-stranded DNA, we have a negative strand and a positive strand. The negative strand Negative is shorthand for antisense, and positive is shorthand for sense. So what that means is the sense strand, that sequence is the same as the sequence that's in the messenger RNA, except of course that we're going to have U's instead of T's in the messenger RNA. So this sequence is hanging out in the nucleus, or perhaps actually inserted into your DNA, and it can be used to make messenger RNA. The negative strand is going to serve as the template, and this is the messenger RNA that we're going to get from transcription. We get AUG, that's our start codon, CGA, etc. Using our little decoder here, we can figure out the actual sequence of amino acids, the protein that we get. So we have methionine, arginine, serine, UAG codes for stop. Again, an actual protein would be much, much longer than this. But your cell is going to provide all the enzymes, all the equipment, the ribosomes, etc., needed to transcribe and translate the entire genome, make all the proteins needed to make this herpes virus, assemble those proteins, put them together to build the capsid, etc., and then the virus can go on its merry way and infect another cell. The next group I've decided to look at are the plus or sense single-stranded RNA viruses. And my main reason for picking this group is there's a member that I'm sure you're well aware of. COVID-19. We all know COVID-19, of course. COVID-19 contains sense RNA. Sense RNA can serve directly as messenger RNA. So sense RNA is something the ribosome can make sense of. If you are infected with COVID-19, it inserts its genetic material, and that can be used immediately by the ribosome to spit out viral proteins. Now remember, the viral proteins are going to form the capsid, the package. But we still need copies of the RNA to put into the container. So that RNA has to be copied. These viruses, these plus single-stranded RNA viruses, carry with them an enzyme that you don't have in your cells. It's an enzyme that can use RNA as a template to make a complementary strand of RNA. So this enzyme will grab onto copies of that RNA plus strand and make the complementary strand, which is RNA negative. And then that enzyme will use the RNA negative strand to make RNA positive strands. These RNA positive strands, these sense strands, will build up in the cell, and they'll be packaged into those protein capsids. And then the cell will fill up with these virions, and they'll leave the cell. They're damaging the cell, of course, during this whole process, and they damage the cell when they leave to go off and find new cells. The last group that we have to talk about are the strangest. These are the retroviruses, and HIV is a member of this group. Retro means backwards or in reverse. And the reason these viruses are called that is because they contain an enzyme known as reverse transcriptase that can use RNA as a template to make DNA. And of course, that's the reverse of normal transcription. So HIV contains sense RNA. And we'll use our same hypothetical sequence here. That RNA could, of course, be used directly by the ribosome. But it can also be used as a template to make antisense DNA. So again, this is an enzyme, reverse transcriptase, that is contained within the virus. The genetic code of the virus, of course, contains the instructions needed to make that enzyme, and your cells will blindly make it. If you have reverse transcriptase in your cells, it means you have a retrovirus infection. Once we have this antisense DNA strand, That can be used as a template to make the sense strand of DNA. And these two strands stay stuck together, and now we have double-stranded DNA. This double-stranded DNA is going to hang out in the nucleus, and it may even become incorporated into your DNA. That's the integration part. The DNA can be used in the normal way now. It can be used to manufacture our sense RNA, and that's the same sequence that we started with. So we'll have this transcription occurring in the nucleus, and the sense RNA will head out to the cytoplasm. Some of it will be used by the ribosome to make viral proteins, and some of it will be simply used as the genetic material for the new virions. So the ribosome will translate this, it will make the capsids will fill up with additional sense RNA, and then as the capsid leaves the cell, it rips away part of the membrane of the cell with it. That's a pretty neat trick too. So we've talked about a few other enveloped viruses, but by ripping away part of the outer membrane of the cell, they can use that to kind of hide themselves, mask themselves within your body. Your immune system might have a hard time recognizing that virus as foreign because it's coated in basically the skin that it ripped off of a cell. Before we leave viruses, I have one final question. And personally, as someone who's really keen on biology, I think this is one of the most interesting questions in biology. Where did viruses come from? Well, you might be thinking to yourself, maybe they are a simple form of almost life. Maybe they are related to the first cells that appeared. So maybe they're pre-cells, not quite cells. They're a leftover from 4 billion years ago. That would be a pretty sound hypothesis, but actually the truth is far more interesting. In our cells, we have bits of DNA that are known as transposons. This is a short piece of DNA and it codes for a protein. And the protein it codes for, all it does is cut that piece of DNA out and move it somewhere else. So we have a transposon that codes for the enzyme needed to move the transposon to another piece of DNA. These are sometimes known as jumping genes or selfish DNA because they reproduce on their own for really no reason, just because they can. They move around in your genome and occasionally cause problems. So occasionally they will insert themselves into a gene and turn that gene off. I mentioned that we have a lot of junk DNA. Most multicellular animals do. And in part, that's because of transposons jumping around and messing everything up and turning off some old genes that fortunately we had duplicates of. It's kind of a mess. And it's thought that these jumping genes gave rise to viruses. So imagine adding a few more genes. Even if we just added a couple of genes that would allow for the creation of a protein coat by the cell. Now we have something that's more durable. It can be even more selfish. So this thing exists because it can. It exists because it's good at tricking the cell it's in to make copies of it. So multicellular animals appear to have given rise to these parasites. They're bits of their own DNA that are now parasitizing them. What's even weirder is there are retrotransposons. There are transposons that can code for enzymes that will transcribe them and then copy them back into DNA and insert them somewhere else. Really a fascinating topic. I would be rather remiss if I did not talk about COVID-19, the virus that's been causing us so many issues over the last few years. The name comes from coronavirus disease. The 19 specifically comes from the year that it was first discovered. Corona means crown. You can see the crown on a bottle of Corona beer. So the virus has a crown of spike proteins that help it gain entry into a host cell. So those spike proteins will stick to receptors on your cell and induce your cells to take in the virus. Somewhat interestingly, the sales of Corona beer went up considerably during this outbreak. COVID-19 virus is an enveloped virus, and that envelope is studded with these spike proteins. The envelope, once again, came from the cell. It's made of phospholipid. That's what cell membranes are made of. Part of the genome in the virus is used to code for those proteins. So what happens is your cells are reading that information, they're making those spike proteins, they're being shipped to the surface, and then as the virions leave, they rip away some of the membrane, and that membrane is studded with these spikes. The spikes are used to gain entry into the next cell. Now, the spikes are not something you would normally have on the surface of your cells. This is something your immune system could potentially recognize as unusual. Your immune system makes antibodies that stick to unusual proteins. So antibodies could stick to these, and then that identifies these spike proteins to white blood cells. White blood cells recognize that this whole thing is something weird, and hopefully they'll try to destroy it. Those spike proteins, because they're unusual, have been used to produce vaccines against COVID-19. And one of those vaccines is known as a messenger RNA vaccine. What they do here is they take the gene out of the virus that codes for the spike protein, and they put that messenger RNA that codes for that spike protein into your cells. And your cells manufacture that spike protein, ship it to the surface. Your immune system recognizes that as unusual, but the vaccine does not contain anything else. It doesn't contain the information needed to make the rest of the virus. Over a very short period of time, several vaccines were developed for COVID-19, and these were messenger RNA vaccines. Messenger RNA vaccines have been in development for quite a long time, contrary to popular belief. This was the first time they were used on such a large scale, though. So with a messenger RNA vaccine, what you do is you take messenger RNA that codes for a small part of the virus, you put that into a little packet that can be injected into the body and taken up by cells. And that was the big stumbling block, figuring out a way to get this messenger RNA into cells without it being destroyed first. Once this viral messenger RNA gets into your cells, your ribosomes will blindly decode it. And again, that's because the code is universal. They're going to make a protein out of that. And the messenger RNA that has been injected codes for the spike protein of COVID-19. The spike protein will move to the surface of the cell and it will be displayed. Your immune system will recognize this protein as foreign and produce antibodies against it. Now, let's say you actually encounter the real virus. That virus will have spike proteins on it, of course, and these antibodies will stick to it. So your antibodies will stick to the spike proteins, and that will stop the virus from attaching to your cells. It also identifies the virus as something foreign, and your immune system will attack it and destroy it. Unfortunately, there have been a number of misconceptions as to what the point of vaccination was, and what the point was to self-isolating and wearing masks and social distancing. Let's start with the vaccine. If you are vaccinated, you can still get sick. That's the case for any vaccine. You can still take in the virus. You can still breathe it in, of course. We're not going to stop that with a vaccine. And it can still damage some cells before your body is able to respond to it. If you have been vaccinated, your immune system gets a head start. You already have some antibodies flowing through your blood. You already have a population of B cells producing those antibodies. If you haven't been vaccinated and you're exposed to a virus, it will take some time for your body to make the antibodies and just get started launching a response. If you've been vaccinated, you will be able to get rid of that virus much quicker. It still might create some symptoms, but the symptoms will be greatly reduced. And more importantly, in the case of COVID, you will be infectious for a much shorter time period. The period in which you can spread it to other people will be greatly reduced. If you're wearing a mask, that is going to help protect others. It protects others more than it protects you. When you cough, you are releasing great big droplets of fluid, and those are going to get stuck in the mask. The air you're breathing in already contains some viruses that are basically just tiny particles floating in the air. You might be breathing in some viral particles, but you're not shooting out too many of your own. And with social distancing, of course, again, you're less likely to infect other people. But the whole point of all of these things was never to protect healthy people. Healthy young people are unlikely to have serious side effects due to COVID. But there is a segment of the population, the elderly, people with coexisting infections, people who are morbidly obese, have diabetes, etc. These people are far more likely to be affected by COVID, and those are the people we were trying to protect. The other thing is, and you've probably seen this before, this whole idea of flattening the curve. The idea was that if we social distance and do all this, the disease will spread more slowly. It's still going to infect a lot of people. It's actually going to be around longer because we're delaying these infections. But we never get to a point where we overwhelm the hospitals. And that was key, especially in the beginning. If we had just let things run their natural course, everyone would have got infected very quickly. The infection would have been over faster, but people would have got infected very, very quickly, and the hospitals would have been overwhelmed. We would have seen a lot more loss of life, not just from COVID patients, but from other patients that couldn't be admitted to the hospital because the hospitals had reached their capacity. Did these measures save lives? Most definitely. I think everyone can agree on that. What's more debatable is how many lives were saved. It's thought that close to 7 million people died of COVID-19. Depending on who you talk to and where you get your numbers from, it's a very difficult thing to estimate. There may have been somewhere between 10 and 20 million lives saved. Now, it is very difficult to estimate this because people that died of COVID-19, for the most part, had other problems. They had other issues, but COVID-19 was the last straw. These are vulnerable individuals that couldn't deal with COVID-19 as well. Is it worth saving 20 million people? I think so, but not everyone does. This is kind of a personal thought process that you have to go through. There are roughly 8 billion people on the planet. 20 million is a fairly small percentage of that. It did have big impacts on the economy of the planet. It increased rates of depression and so on. So it is a very tricky topic to address. I think it was quite amazing that all the countries of the world came together to stop the spread of this disease. Microbiologists, pathologists were waiting for something like this. This was not entirely unexpected. We had an outbreak of the Spanish flu back in 1918 that killed 25 million people. Population of the planet was quite a bit lower then, so that was a significant chunk. And we were just waiting for something else like this to happen. These kind of respiratory diseases pop up every few decades. A serious one pops up every 50, 60 years or so. So yeah, it's quite interesting that everyone managed to work together on this. We did save some lives. It's going to be interesting seeing how this pans out going forward with other outbreaks. The precautions taken to limit the spread of COVID-19 were definitely a major inconvenience for just about everyone and a major hardship for some people. Personally, as someone with elderly parents who were in a very high risk group for this disease, I myself was happy to make those sacrifices. But again, it does come down to a person by person choice. The one thing I am quite concerned about is we definitely will have more infections. We will have worse infections than this. There were a lot of misconceptions and a lot of misinformation spread during this period. And I'm hopeful that those misconceptions won't limit the effectiveness of future vaccination and preventative measures. And now for something that might be even more fascinating: prions. Prion stands for proteinaceous infectious particle. And this is an infectious agent that's far simpler than a virus. This is an infectious agent that doesn't have a genetic code, which is bizarre. I can't think of any other entity that can reproduce that doesn't have a genetic code. Even a computer virus has a code. These are proteins that can bind to other proteins and change their shape. They're responsible for a few rather rare and poorly understood diseases. This poor sheep that you're seeing here is suffering from scrapie. Now, it looks like this disease is causing fur loss or wool loss, I guess. But actually, that's not the case. What's happening is this proteinaceous pathogen is found within nerve cells that are found just beneath the skin. So peripheral pain receptors, for instance. And they're damaging those cells, interfering with how they would normally function. And this poor little sheep is in quite a lot of pain. So its skin is burning. It feels like it's on fire. And what they do is they rub up against walls and fences and basically rip the flesh off of their body. Prions are responsible for bovine spongiform encephalopathy, which you probably know as mad cow disease, and the human variant of this, which is Creutzfeldt-Jakob disease, CDJ. Alrighty, so we have a protein that has gone rogue. It's got an abnormal shape, and when it comes into contact with cells, it damages them. That doesn't sound too out there. That doesn't sound too unusual. It sounds a bit like it's acting like a toxin. However, I told you that this abnormal protein can reproduce. How the heck can that happen? Well, let's take a closer look at that scrapie example. What you're seeing on the left is a protein that's found on healthy nerve cells. This is its normal configuration. On the right, you're seeing an abnormal form of this protein. There's very little change in the sequence of amino acids. Instead, it's being folded differently. And you can see it has a very different shape. And as you might imagine, it can't do the same things that the normal protein does. But this is where things get weird. This abnormal form of that neuronal protein will grab onto the normal protein whenever it encounters it, twist it around, change its shape to match itself. Imagine if you could do this. Imagine if you could go out on the street, grab a stranger, grab his face, twist it around until he or she looked just like you. After a while, everyone would look like you. Is that reproduction? I would say so. I mean, you're making copies of yourself in a really unusual way. You can imagine that if this abnormal protein is found within nervous tissue, after a while there might not be much of the normal protein left. The abnormal protein will replace the normal protein. But there's more to it than that. because the abnormal proteins stick to each other. So not only will the abnormal proteins replace the normal proteins, but they'll stick together in long chains and form these plaques or deposits that will cause further harm to the cell. Now there's one more thing that's really weird about this, if it wasn't weird enough already. This is perhaps the only disorder or disease that is both heritable and transmissible. Something that's heritable is something that involves genetics. It's something you inherit from your parents, or perhaps during your creation, there was a mutation in the DNA that combined to create you. Transmissible refers to the fact that we're dealing with an infectious agent. So if you get the flu, it's because a flu virus was transmitted to you. Well, prions can show up in your body by either pathway. Let's say you have a gene for this particular protein. Remember, it's supposed to be found in its normal configuration on your nerve cells. Let's say you have a gene for that protein that is mutated. And it's mutated in such a way that you get the abnormal form. Well, if that happens, you're going to have problems. But alternately, let's say that you consume some nervous tissue, and that nervous tissue contains these prions that are misshapen, and they travel to your nervous system, grab onto the proteins that are found there, the normal proteins, and change their shape. You would end up with that same unfortunate condition. Now you might be wondering, how would you ingest nervous tissue? It's not something that most people consume. Well, let's say you're eating a T-bone steak. That T is part of the vertebrae. And as you've probably noticed, there's a little hole that contains a little bit of soft tissue, and that's the spinal cord, and it could contain prions. Now, I should say it's incredibly unlikely to contain prions. This is a rather rare, as in extremely rare, condition. Or it could be that you're eating some sort of ground meat that contains a little bit of nervous tissue. So what do prion infections do to you? Well, the name for one of these infections is transmissible spongiform encephalopathy. And let's break that down. So encephalon means brain or head. Pathology, of course, means a disorder or disease of, so something is abnormal. Spongiform means having the form or shape of a sponge. A sponge, of course, has lots of holes in it. So we're talking about this transmissible pathogenic agent that can cause your brain to become like a sponge, to fill up with these little microscopic holes. And that's what you're seeing in these histological sections. So you can see there's these white areas where there should be cells, but there's not. That normal protein is important for maintaining healthy cells. It's not there anymore. And in addition to that, the abnormal proteins have linked up to form these harmful plaques. And the cells in that area have died. We have BSE, bovine, which means cow, spongiform encephalopathy, and then we have the human form of that disease, Creutzfeldt-Jakob disease. And there's some debate as to whether or not these are distinct variants or whether they're actually the same thing. We have scrapie that we've talked about, and a new one here, Kuru. Kuru is rather interesting. It's something that's exceedingly rare. It was a bit more common in the past, and it was most common in natives of New Guinea that were still practicing forms of cannibalism. They would eat the brain tissue of ancestors and enemies, and they would pick up the prions, and the prions could pass through the digestive system, pass through the circulatory system, and infect their nervous tissue. These are terrible diseases. They cause a wasting away of the brain, and there's nothing you can do about it. In Kuru, for instance, people that have this quite often stop sleeping. They don't sleep for a month or more before they die. They laugh uncontrollably. They basically have a mental breakdown, which is rather unpleasant. more and more prion diseases are being identified, not just in humans, but in other animals as well. But again, note that they are quite rare and also very poorly understood. We have a few different forms of human prion diseases here. You can see they affect different parts of the brain, and that's because we have different proteins that identify those different areas of the brain, and those proteins can go bad. Well, that was a rather long topic and a rather technical one at that. Very quickly, going over the concepts, you should be able to explain to me why viruses are not generally considered to be alive. Again, that's something you can get into a debate about depending on who you ask, but most biologists will say, no, they don't quite fit the definition of life. Why is that? Some viruses contradict the central dogma of molecular biology. Remember that this tells us that DNA is the long-term repository of genetic information. It's used to make RNA, which is then used to make protein. But some viruses break those rules, and you should know which viruses do that and how they do that. Viruses are obligate parasites. They don't have a metabolism of their own. They can't do anything. If they're outside of a cell, they're completely dormant. They do nothing at all. They need to hijack a cell in order to get anything accomplished. Some viruses can integrate their DNA into the DNA of the host cell, and that results in a latent infection where we have a provirus in the genome of the host cell. And things can stay like that in a dormant state for decades. Viruses that are enveloped steal plasma membrane from their host, and also any proteins that they have had shipped to the surface of the cell. And this coating of plasma membrane can help them avoid detection. Fortunately, though, enveloped viruses are quite vulnerable outside of the body. Soap and water is enough to disrupt that lipid membrane, and if you do that, they can't enter other cells. Prions are proteins that have changed their shape, their conformation, and they can bind to similar proteins and change their shape. It's really quite bizarre. Here's a short little terminology list for you. Again, watch out for these terms on your midterm and final exam. And finally, a few study questions to think about. Also, if you need a little bit of help with that molecular biology stuff, or you want a little more background on it, I do have a video on that. So follow that link here for that. I do have a longer video on viruses as well. If you're really interested in this, you can look that up on my microbiology playlist. All right, after this, we're going to get into actual living things. And we're going to start by looking at the simplest living things, the bacteria.